Hello. I'm Jason Linville. This lecture is being recorded for CJ352 Forensic Science Biology Lab 2. This is the lecture on mitochondrial DNA, and I'm going to divide it up into 3 mini lectures. So this one specifically is on background of mitochondrial DNA. So we're going to review a lot of the stuff that we've gone over in 110, and then we'll get into the new, fun, confusing details stuff. Okay. So we want to review what the structure of DNA, the packaging of DNA, that means not in chromosomes is what I mean for packaging. So we're really going to contrast mitochondrial DNA with nuclear DNA, including the total length of mitochondrial DNA, which is going to be around 17,000 compared to 3 billion. And then also the length of the area that varies among humans. You know, and we talked about nuclear DNA with the STRs. You can pick out, there's a whole bunch of STRs loci in the human genome that you could select for individual identification. There's not as many choices with mitochondrial DNA. In fact, there's one choice. So we're going to go over that. And then finally describing why mitochondrial DNA from some samples, this means like an evidence sample, like a cut piece of hair, might be able to produce mitochondrial DNA, enough mitochondrial DNA for analysis, but not nuclear DNA for STR analysis. And then, you know, the disadvantage of mitochondrial DNA. And then finally, the one new thing that we're going to do that's different from 110 is we're going to actually look at family trees and talk about how to read family trees and trace maternal relatives. Okay, so that's going to be a skill that I want you to do, where if I show you a family tree, that will be on the quiz. Oh, crap. How? Because you can't color it in. Oh, okay. You just have to write out the numbers. Sorry. I'm identifying problems and solving them in real time. Okay. So mitochondrial DNA, the nuclear DNA, right? The 13 STR loci, 13 pairs of numbers, they all come from, if I would remember to change my mouse to a pen here. Okay, they all come from the nucleus, this area right in here. That's where, even though it's just squiggles here, you would find 23 pairs of chromosomes if you cracked open the nucleus. Okay, the DNA that we're talking about now is mitochondrial DNA. It's in the powerhouse of the cell, these mitochondria. The reason why mitochondria have their own DNA is, the theory is these used to be like little unicellular organisms that could live on their own, and then they got engulfed by a larger cell, and they lost their function and formed a symbiotic relationship with the cell, where this produces ATP, energy, oxidative phosphorylation undergoes in here, and it sort of supplies that to the entire cell. So there's a couple of things here. Number one, oh, well, if it's historically was a single cellular, was its own cell, then that explains why it has DNA there. The other way you can look at it is it would be extremely inefficient for all of that, because the mitochondria needs a lot of proteins to go in the membrane to help transport electrons. It would be extremely inefficient if you coded for a protein the mitochondrial needed here in the recipe book called the nuclear DNA, and then made messenger RNA that went over to the ribosomes on the endoplasmic reticulum, and then the protein somehow had to be transported into the mitochondria. That's a lot of transport for those proteins. Instead, it's much more efficient. The mitochondria codes for what it needs, makes what it needs, and uses what it needs all inside that mitochondria. Okay, so therefore, mitochondria has its own DNA that codes for the proteins, the little nugget machines that the mitochondria needs. Okay. Mitochondrial DNA is a double helix. So we can think of it as a twisted ladder. We can think of it as a ladder. It still is composed of A, G, C's, T's, and an A is opposite of T. Excuse me, a G is opposite of C. That's what I get for chugging cheap soda instead of water before the lecture. All right, so if we zoom in on an area, just like for STRs, we see GATA, GATA. Okay, for this, when we zoom in, we're also going to see those 4 letters in a sequence. Now, it's not going to be repeated. That's going to be one of the differences, but we are going to see those same 4 letters, and we're going to talk about the sequence of those 4 letters. Mitochondrial DNA is not packaged into chromosomes. So you don't get one from your mother, one from your father. Okay, it's not paired chromosomes. It's just one big ring. So again, this is a double helix ring, but it's just sort of spiraling around. And then it's like if you took this end and connected it, wrapped it around and connected it to that end, so it's in a big circle. Okay, so you take the double helix, the long double helix, connect it into a ring, and that is your mitochondrial DNA. So again, double helix, yes. Okay, composed of A, G, C's, and T's, yes. Chromosomes, no. Pairs, no. It's just a ring of DNA. Okay, so let's talk about what, if we actually looked at the letters in this ring of mitochondrial DNA, what we'd be looking at. And this is, oh, I should add, this is human mitochondrial DNA, okay? Because other animal mitochondrial DNA is going to be a little bit different here. Okay, so in human mitochondrial DNA, this ring, if you start right here and count all the way around, it is about 17,000 letters long. Okay, so 16,569. Some individuals may have a substitution or a deletion or two, but that's pretty much a good number for everybody. And so that's reasonable. Like it's no longer 3 billion letters, it's 16,000. Feasibly, we could think of, oh, well, if you're analyzing mitochondrial DNA, it's probably a printout with 16,000 letters on it. And then you just compare that between your evidence and your suspect or whatever. Not exactly, because like we talked about in the nuclear DNA, most of the DNA, 99.8%, is identical among humans. In mitochondrial DNA, it's a little bit smaller. It's more like 98.3, okay? But it's still, in certain areas of mitochondrial DNA, in the genes that code for the proteins we need, we can't be different. Okay, I can't have a cytochrome B that's different from Jesse's cytochrome B. It needs to be the same because it does the same function. So starting here all the way around this ring up to right here, we're going to be exactly the same. Like if we took everybody in the class and sequenced all 15,700 of those letters, we would be exactly the same. Okay, maybe like one base pair difference between a couple people, but we're all exactly the same. However, in this D-loop region, okay, in this D-loop region, there's 900 bases, and in those 900 bases, there's that 1.7% difference. So I actually, my previous number was a mistake, because here we're 100% similar. It's just in this D-loop that we're 98.3% similar. Okay, so we could probably do the math to figure out what that total percentage is, but anyhow. Okay, so it's just in this D-loop. This D-loop does not code for a protein, which is why there's free to be mutations among individuals. It does contain a center area that initiates replication. So it's sort of like the beginning of where when you need to copy your mitochondrial DNA naturally when your cells are dividing, it starts at that origin right in the D-loop and then wraps around. So it's sort of like the beginning, there's some flexibility, and the end, there's flexibility, but all those genes in between are exactly the same. All right. Okay, so to review, double helix, yes. 46 chromosomes, no chromosomes, just a ring. All right, now this is the big thing of why forensic scientists actually care about mitochondrial DNA and why it can be useful in some situations. It's one of the reasons. There's actually two reasons. Okay. In nuclear DNA, there's only one copy of each allele per cell. Okay. So in other words, if we're trying to analyze TPOX and you're on 1011 at TPOX, you have in one cell, you have one copy of that 10 allele, okay? Areas on whatever chromosome number 3, whatever it happens to be, repeated 10 times. And one copy of the 11 allele from your mother or father, the other one, okay, repeated 11 times, just one copy. In theory, we could take that one cell with that one copy and we could amplify those alleles just like we did when Simone was mowing my yard, okay? 1 to 2, 2 to 4, 4 to 8, etc. But when you're dealing with only starting with one copy, the amplification reaction usually never takes off. In order to get reliable amplification, you really need about 300 or so copies of DNA in your extraction, in your amplification reaction. Okay, so like for one cell, you're probably not going to be able to amplify DNA from that one cell, okay? All right. For mitochondrial DNA, there's multiple copies of mitochondrial DNA, meaning there's multiple rings in each mitochondria, and there's multiple mitochondria in each cell, okay? So there may be more than one mitochondria in each cell. So let's take this situation. Let's say we have a forensic science sample, okay, and it is like a hair. It's not a hair with a hair root that has a lot of cells in it. It's just a hair, and it essentially has the equivalent of 3 cells from the person embedded in the hair. Okay. So if we have a forensic science sample with 3 cells, we have 3 copies of allele 10. Okay. I don't know what I'm drawing here. Okay. Allele 10. And we're starting off with 3 copies of allele 11. Okay. In the nuclear DNA. And like I said before, you need about 300 to take off. So whether we have 1 cell or 3 cells, we're not going to be able to amplify short tandem repeats. Okay, you can attempt to. You can put the primers in there. You can put the DNTP, Taq polymerase. You can put your DNA, the maximum amount. But if you're only squirting the equivalent of 3 cells worth of DNA in there, the amplification is probably not going to take off. But how many mitochondria do we have? Well, let's say that we have 10 mitochondria in each cell. Okay, all of a sudden, 10 times 3 equals 30. Wait, I think you guys can probably, there we go. Let's give myself a little applause there. All right, since 10 times, that means we have 30 mitochondria. Okay, let's say there's 10 copies of mitochondria, 10 rings in each of those mitochondria. Well, 10 times 30 is 300. So in 3 cells, we may have not enough nuclear DNA to amplify and analyze short tandem repeats, but we do have enough of those mitochondrial rings where if we do the same thing for mitochondrial DNA, squirt our DNA in, and now it's 300 copies plus primers plus DNTPs plus polymerase, we can amplify mitochondrial DNA. Okay, so that's the big advantage down here. Mitochondrial DNA can be used for older degraded samples when STRs fail. Okay. What do we look at for mitochondria? Well, we're going to get into the details of how this happens, but what I want to emphasize is for nuclear DNA, it's the number of repeats. We are not looking at repeated regions of DNA in mitochondrial DNA. We're taking, it's only 900 base pairs long. We're just looking at the sequence, okay? So in other words, a genotype is not going to be a profile that goes into CODIS has 13 pairs of numbers. Okay, a profile that would go into a mitochondrial DNA database or for mitochondrial DNA would have 900 letters. Now we're going to find it's not actually that, but that's how I want you to think about it. Okay, it's just the 900 letters in that region, that D-loop region that we're going to amplify and analyze. Okay, so we look at the sequence. And if you remember all the way back where we talked about how DNA can be different, we're going to look at, the way sequences are different. You know, a G changes to an A, okay? There could be an addition of 1 letter. It's no longer that repeated DNA. Okay, so uses of mitochondrial DNA. Help identify the source of degraded evidence if STRs cannot be amplified. That's what we just talked about. Okay, also another use of mitochondrial DNA and sort of advantage is it can be used to identify human remains by comparing DNA to a maternal relative. Okay, now this relates, here's some tips for your mock case questions due at the end of the week. A couple of them are where you find human remains, you know, dead body in the woods, and you're trying to identify it. The best way to identify a body is, well, the body is the evidence, right? So the evidence is the unknown sample. You just get a sample from the known suspect, right? You just compare it to the suspect's DNA. Well, here we don't have a suspect. We're okay. Let's say I turn up missing and the human remains. We want to prove that it's Jason Limbaugh. Okay, it matches my biological profile from the anthropologist, but we want to confirm it. All right. Well, all you have to do is come and get DNA from Jason Limbaugh. But wait, I'm missing. Okay. They think that you can't take a sample from the body. It's self-fulfilling prophecy, right? But can you guys think, go ahead and throw in the chat box, because this is going to be an answer to one of those mock case questions. If I turned up missing today, right? Okay, and then 3 months later, they find badly decomposing human remains in Oak Mountain, you know, like one on a hike and got lost or something. Okay, so they get, what is the best way? If they get DNA, STRs, because you have human remains, you can cut into a bone and still get STRs. Okay, if you get STR profiles from that body and you want to compare it to my DNA, where could we get my DNA? I've been missing for 3 months. Is my DNA anywhere that we could access and mention it? Yeah. Okay, so we have 2 answers here, right? All right, Becca says a hairbrush. And that's good because that would be my DNA, right? Okay, it would be my DNA from a hairbrush, and then we can compare 13 pairs of numbers from the hair in the hairbrush because it has hair roots. Okay. Assuming let's pretend I have luxurious hair that I brush 100 times every morning. Okay. So we get DNA, 13 pairs of numbers from the hair, 13 pairs of numbers from the body. We can say, yeah, this body is Jason Linville, no doubt about it. That's what you want to try to do first to get that STR profile. Okay. But yeah, you also have to double check the hairbrush. Hey, if I had luxurious hair and brushed it 100 times every morning, no one would be allowed to use my hairbrush. It would be blue. All right. But what if I've been missing for 2 years? Okay. What if my family has thrown my beautiful blue hairbrush in the dump and there's no longer any DNA from me? Well, then we could go to my kids or parents, but we couldn't get those 13 pairs of numbers necessarily, especially if I only had a few relatives and these are human remains that we're trying to identify from like 100 years ago. What we would do is we could trace the maternal line because mitochondrial DNA for maternal relatives is going to be identical. Now, it's not going to be a definite match here, or excuse me, we're not going to be able to come to a definite conclusion because there are other individuals with this mitochondrial DNA. But in cases of a missing person where you want help identifying the body to make sure it's me and not just some random person, using mitochondrial DNA is a possibility. If you remember, I think we went over this briefly in 110. And here's a graphic picture again, okay, sperm entering the egg, 13 pairs of numbers, 13 pairs of, I'm all up on my DNA profile. 23 chromosomes in the sperm, 23 chromosomes in the nucleus of the egg, and boom, they come together, and now you have 23 pairs of chromosomes. Okay. The sperm cannot carry mitochondrial with it. So notice the mitochondrial that you have all is in the egg of the female. So like I may have said in 110, it doesn't matter how masculine you are. I, for one, am very masculine. I took a test. I will be happy to share the results with you at some time in the future. But no matter how masculine I am, I still got all of my mitochondrial DNA from my mommy. Okay, none from my dad. My boys, my children, my beautiful baby boys. They don't have my mitochondrial DNA. Okay, because this is me right here. Is this getting even more graphic? Okay, and this is my wife. All of their mitochondrial DNA came from their mother, which is probably why when we see people on the street, they go, oh my, what a lovely child. It looks like he oxidatively phosphorylates his molecules just like his mother. Okay, I'm going to start saying that too. I'm warning anyone with children. That's going to be the next thing I say to you when I see your children. Okay. Mitochondrial DNA from the mother. So that means, okay, I have the mitochondrial DNA from my mother. I have the mitochondrial DNA from my sister because we have the same mother. I have the mitochondrial DNA from my brother because we have the same mother. Okay, again, assuming that we have the same mother here. Okay. My sister has children. Okay. My nephews, Keegan and Nolan, they have the same mitochondrial DNA as my sister, Kristen. I'm giving you guys some, okay, who has the same. So I have the same mitochondrial DNA as my sister's children, whether they're boys or girls. Now, my brother's children, okay, they have the same mitochondrial of that person that he married. So it's not my brother's, it's not my mitochondrial DNA or not in my brother's children, okay? All right, so that's how this maternal works, okay? So it's not specific to, nice try, Rebecca. Becca's trying to hack into my Minecraft account and destroy my home. This is a true story. No, I'm not going to put this down on recording. I'm building a house in Minecraft that's exactly like the house. Sorry. All right. So mitochondrial, so if we're using it as a crime or to identify a body, yeah, it might be me, but it also may be my cousins or my maternal relatives. But luckily, they probably haven't gone up missing. But if it's a crime where there's, for example, if there's a hair on the dock on a boat dock with some hair in the pliers, okay, and we can only get mitochondrial DNA, we can say, well, it's consistent. It matches the mitochondrial DNA from Lacey Peterson because we got her toothbrush, okay, or got it from her mother. But it could also be any other maternal relative of Lacey Peterson. It can also be a random individual who's not related, but just happens to have that same mitochondrial profile. Because again, we're just looking at 900 letters. There's not a whole lot of information, or there's not a whole lot of difference. So it can be where your maternal relatives will match your mitochondrial DNA and a random stranger might match it, okay? The Casey Kaylee Anthony case, okay? We found DNA in the trunk of Kaylee Anthony's car, but it doesn't mean that it's the child because it could also be DNA from the mother. So it's not as strong evidence, okay? So that's the disadvantage of mitochondrial DNA. It's not specific to an individual. Could be random, could be maternal. Okay, so let's look at these trees. You guys can treat and flash ahead. All right, so we're going to ask, Elise, are you present? You can participate either in chat or at a microphone. What I'm going to ask you to do is, this is how we read the family tree, okay? We have a male, number 1, and a female, number 2, and they had children. We follow the line. They had a son, 3, okay? And they had a daughter, 6, okay? The daughter, number 6, married, because this is a proper family tree. I could care less, okay? Male, number 5, okay? Son, number 3, cohabitated with, I don't know, giving a backstory to shapes and numbers, okay? And number 4. Okay, and I'll get to your question, Marla, after we've finished the first example of family tree. Okay, so the question here, and then they had 2 children, and 5 and 6 had 2 children. My question is, Elise, who in this family tree would have the same mitochondrial DNA as grandma number 2? Okay, and this is going to be the question on the quiz or exam. Okay, I'm going to show you a family tree, and I'm going to say list the numbers of individuals who have the same mitochondrial DNA as that number. Okay, what do you guys think? Actually, maybe I shouldn't pick on Elise. She maybe have strolled away from her computer. If anybody wants to write in the chat box, let's take multiple guesses. Okay, tell me who is going to have the same mitochondrial DNA as number 2. All right, so Tabby comes in with 3, 6, 10, 3, 6, 9, 10. Okay, 3, yes. 6, yes. 9 and 10, yes. Because remember, 6, it doesn't matter if they're male or female, they're both going to have it. Just like number 2, it didn't matter if they were male or female, they're both going to have it. Okay, but here, it's not going to be 7 and it's not going to be 8 because they're going to match number 4. Okay, so does everybody see that? And let's fill it in on the next slide. Do I have them colored in? No. Oh, you know, I changed the color scheme for this. And if you can tell, these colors are slightly darker green than the other almost identical green. I need to go back and change the colors again. Okay, anyhow. So yeah, you guys are correct. 2, 3, 6, 9, and 10. All right. Now, again, I will show you this. I won't always give you 2. I may say, who matches number 10? Okay. Well, if number 10 is on that line, then it's going to be the same answer, right? It's going to be 9, 6, 2, and 3. Okay. But where you have to watch it, if I say who matches 7, know that you're not going all the way back up here. It's just going to be 7, 8, and 4. Okay. So you should be able to see this tree and be able to identify who has the same mitochondrial DNA. All right. Let's go with a slightly more complicated one. Here we have 1 and 2, except we have 3 kids, son 3, daughter 6, daughter 8. They had children here, the grandkids, and then we have one more generation in there. All right, so I'll ask you to start out with the same question. Who has the same mitochondrial DNA as number 2? So go ahead and think about that and type it all in. Somebody give me an answer in the chat there. I wonder if we're on maybe on a slight delay here. Actually, it takes a while, I guess, to get all these in here. It's 2. All right, here we go. We got the first one in here. Okay, so we got 2, yeah, and it's going to match 3, 6, and 8. Very good. And then 11 and stops right there, and 13 and 14. Or whoops, sorry, 11 and 14. You guys are right about that. Okay. And then finally, we don't go down from 11, but we do go down from 14 to get 16 and 7. Wait a minute. Do I have, what is going on here? Yes. So 6 and 7 or 16 and 7 would have been correct because for some reason these got incorrectly identified. So yeah, you guys got the general idea. So again, the biggest issue I have when I give this question, or not issue, but the most common mistake is if I have you start down here, like at 14, then you'll get the 6 and the 7, and you'll get the 8, and you'll get the 2, but people forget to go across and hit all the siblings at each level. Okay. So you got to be careful about that. All right. Just like I will try to do a better job of actually typing in real numbers in the future. Maybe I needed to enlarge. I bet I needed to enlarge those text box and the letters got cut off. Okay. All right. So that is everything for that first section on just the background of mitochondrial DNA, where a lot of it is review from 110. Not that everybody realized that. And let me go back here and answer why it's not 15. But the extra is this maternally inherited. Okay. The reason, so Elise asked a good question. The reason it's not 15 is because remember, this is the male. So 15 would match 12. So again, it goes down from 6 because it's female. It goes down from 8 because it's male, but it does not go down from 3. Just like it goes down from 14 to include 16 and 17 or whatever the heck these are. But it does not go down from 11. All right, so does everybody see that? Yeah. Okay. But I mean, that's a good question, at least, because it's, I mean, those are the type of common mistakes when you start going through these too quickly. So you always got to pay attention, not just going up and down, but also pay attention to the circles and squares, the males and females. Okay. Marlo asks if, going back to the original statement of comparing it, talked about if there was only a father for comparison. If there was only a father for comparison, then essentially you're out of luck. Well, you might not be out of luck, because let's say that I turn up missing and my mother has passed and my father is there and they're like, we want a mitochondrial DNA profile from your dead wife. And he's like, well, I had her cremated. And they're like, too bad, invent a time machine. I don't know what they're saying. Okay. It doesn't have to necessarily be my mother. It's any maternal relative. So what they could do is if my sister was still alive, then they could access my sister. And going back far enough with your family tree, you could go back like, okay, my mother would have the same one, meaning my mother's mother. Again, my grandmother on my father's side, that does me no good because that's up my father's tree. So my mother, her mother, Grandma Mary, okay, let's see here. She had another daughter, Aunt Sandy. So Aunt Sandy has the same, my mother's sister has the same mitochondrial DNA as me. Aunt Sandy had 2 kids, okay, JG and Joy. So we could, JG or Joy. Ooh, Joy just had Medi. So Medi has the same mitochondrial DNA as me. Boy, the next time I'm at a family, I'm going to bring that up. Think, we had a big vacation where we had everybody together. And I didn't even mention the common mitochondrial DNA that we all had. All right. So let's go ahead and maybe end this mini lecture and move on to the next portion.